So yeah, I'm gonna kind of continue on the theme of not a toxicologist, not necessarily a subject matter expert in MPS specifically. I've got a lot more high-level expertise, but I am excited to kind of share a little bit about my organization, the 3Rs Collaborative, about our MPS initiative specifically. And I am, just for a background, I'm currently executive director of the 3Rs Collaborative. So just a super quick intro to who we are in case you haven't heard of us. We are a US-based nonprofit whose mission is to advance better science for both people and animals. And we really do this by facilitating collaborative 3Rs efforts focused on refinement, reduction, and replacement. We really try to span across the scientific field. We're not just focused on pharma or academia, but we include CROs, government agents, nonprofits, and this is just our board of directors and our leadership team. But we really try to emphasize that collaboration across different disciplines and sectors. We've got 3 core goals. One is about just promoting the 3Rs broadly as a concept and making sure that people are knowledgeable and supportive of it. We then also promote specific 3R strategies that we see that are high impact, evidence-based, and practical, but aren't necessarily maybe being used to their full potential. And then we promote our organization so that people can actually find our resources and our collaborations, etc. I do want to briefly highlight that we do truly work across the 3Rs. We're not just focused on replacement, although certainly that's a very important part of what we do. We do have artificial intelligence and microphysiological systems initiatives, but we also focus on refinement efforts. We have a training course that will be developed and released soon, and we have stuff on culture of care and compassion, fatigue, resiliency. But of course, today I was to talk about our MPS initiative, so that's what I'm going to be focusing on. And this work really seeks to increase adoption and regulatory use of MPS where scientifically justified. Our MPS initiative is unique in that it is primarily composed of commercial developers of MPS who we see as really critical subject matter experts in the field who have platforms that are often more further along in their development than maybe some academic platforms. And since they're widely available, not proprietary to a certain pharmaceutical company, we think they have a lot of potential for much widespread implementation. And we actually do include a couple other nonprofits, consultants, end users. This group is really open to anybody who has an interest in MPS, as well as some what we call enabling companies that are really important for using MPS, even if they may not manufacture an MPS themselves. We've got 3 key working groups. One is focused on the regulatory use of MPS. One is about our technology expo, and that's really about helping connect end users to the technologies that they might use. And the third is about end user engagement. So how do we connect with end users, provide them education, and provide them the information that they need? So I'll just highlight a few of things that might be of interest of you. So we do have this technology hub that connects end users with relevant commercial technology providers. We kind of model this after like a shopping platform where you can filter by maybe like size and color of shirt. But instead here you can filter MPS by tissues or disease type or even species. So that helps you kind of narrow down the companies that might work best for you a little bit faster than just trying to Google them or by word of mouth alone. And then we also are planning on creating a publication hub from all of our companies that highlights their relevant publications. So then maybe you can dive in a little bit further before you proceed with maybe contacting them directly. We also have an MOU in place with the IQ MPS. Essentially what they do is they choose topics for our providers to present on, and then we coordinate getting those providers, those developers and like a workshop and registration. And so we've done 10 workshops. Actually, I think it's 11, and we have one more coming up this year on December 4th, focused on oncology methods. And each workshop basically features technology providers doing these back-to-back 15-minute data-driven presentations. So you can really see what might be the benefits of each platform and their use for each. And we also, these are open to anybody. We invite our colleagues from the FDA that are interested. We promote it to our general newsletter. So if you want to join, we would love to have you if you have a specific interest in them. We also actually record these as well and host them on YouTube. So they're really easy to find on our website if you want to reference them later. We do some kind of general education about MPS. So after seeing quite a bit of misinformation related to the FDA Modernization Acts, we have this briefing with just kind of facts about it, what it actually might have changed after it came out. And so this is something that different companies can kind of point to or learn a little bit more about that act and the state of the science. But the big thing that I was asked to talk about today was this cross-platform project we're doing with the FDA eSTAR. We entered into a public-private partnership or consortium agreement with them last December, and we really have this goal about building confidence in MPS. And we decided to focus on drug-induced liver injury because the models on that are really quite mature, and there is considered a need from the FDA for models that better predict DILI. So what we're doing is we're leveraging our connections with the developers to do this kind of cross-platform project. And our goal is to create kind of a universal protocol and experimental design for evaluating DILI with MPS, regardless of the platform you're using. So this includes things like cell types, endpoints, core reference compounds, and our focus is on acute liver toxicity with small models. We're trying to start, since we're going to have a pretty complex design with the multiple platforms, we're starting a little bit simpler with the experimental design. And then what we're doing is we're going to be running this as a simultaneous and coordinated cross-platform study across commercial providers. And we're going to be working to set up those providers with qualification and set up FDA eSTAR for developing best practice and guidance. And that then they can provide to sponsors about what criteria they need to see, validation data, and reporting metrics. And some aspects of this project is we're going to blind the providers to the actual molecules that they're testing. They're going to be provided by a third party. A third party is going to analyze that data. But the experiments are actually going to be run in-house. So this is going to kind of provide us the best case scenario for each platform, what kind of outputs you're going to find. We might be able to find if there might be some advantages to certain platforms or not, what might be expected variabilities that we'll see, etc. We're still in this process. So this is kind of our current timeline and goals. So right now we do have the specific regulatory context of use defined. We're pretty far along in working on creating a platform agnostic protocol. We've done one-on-one meetings with all of the providers and collected their baseline protocols. So we're both allowing them a lot of flexibility because we want to make sure that their platforms are working in the way that's most optimized for them. But we're also we want to be able to run an experiment where we can actually compare the data across platforms and understand what differences there may or may not be. We're going to actually run this kind of smaller scale pilot project in the spring so that then we can submit a DDT grant to maybe do a more full scale project. Right now, this project is really just operating out of in-kind donations from the developers and other partners. And so we're hoping to get more full scale funding so that we can do a more extensive project in the future. And then, yeah, we're hoping to just look at the results, publish them, and this just will provide a lot more understanding for the FDA about what data is expected, especially with this cross-platform perspective. This is our confirmed COU. We took this from CPATH's CIBM workshop that was conducted, I believe, last fall on DILI. This was considered a targeted enough COU that is actually reviewed by regulatory agencies. One thing that was can be a little bit challenging when thinking about using MPS for regulatory aspects is right now MPS are used a lot for early discovery and screening, and the FDA is less concerned about that part of the drug like development pipeline, and so they really wanted to focus on when they would see the data and what they're looking for. So that's a little bit about why we're using this particular context of use. These are our current stakeholders that are involved. So we've got representatives from ourselves, FDA CDER, CPATH has been a really great partner in this as so far. Graham Marsh as well as Samantha have been doing a lot of work with working on the protocol. We've got 7 confirmed MPS developers that have liver models for DILI specifically that are participating, and we have 2 additional that have been invited. We also recently engaged with NICEDM, Nicole Kleinstruer. I know she's part of the OASIS project that you guys are working on. And so we're excited to maybe be able to connect a little bit with that. And NICEDM's hopefully going to actually be able to provide the blinded compounds and actually do the data analysis. And we also have a few end users that recently got involved that are going to be helping us choose which drugs we're actually going to use as reference chemicals in the experiment, and just generally providing feedback on what's developed, making sure that we have buy-in kind of early on. So yeah, we're still in early-ish stages of this project. It definitely takes a lot of coordination among all the providers, etc. But there's been a lot of good learnings, and we're excited to kind of kick it off more and more as the months go on. And then I just want to highlight 2 other upcoming projects in case they're of interest of y'all. One is that, well, it's not quite published, it's actually in press, it was just accepted, is we do have a paper about using MPS for 3Rs and regulatory applications, nuances, stakeholder responsibilities, and perspectives that will be published hopefully pretty soon. It really outlines how MPS can be used for each of the 3Rs and clarifies the definitions, because we see a lot of, from a 3Rs definition, confusion about terminology, as well as where each stakeholder really lands into what they can be doing to advance the use of MPS in regulatory applications. And then what the other project that is very early, just in the ideation stage, is working on kind of a benchmarking survey and/or paper collating all current use of MPS in regulatory submissions, because we don't actually see a whole lot of use currently. The FDA says they're not hearing of that many applications including them, but what we do hear of is maybe one case study here, one case study there, and we'd like to put it all in a paper and kind of see where is it being used, where is it not being used, and then use that to see how things may change over time. And with that, I'll definitely take some questions here. Also, you can always email me if you want to connect with me or have a follow-up question. And then this QR code should help you sign up for our newsletter, which is really the best way to stay up to date. That's where we send out things like our invitations to like our workshops, our MPS workshops, and all our other 3Rs related things. And then if you do want to check out that Tech Hub or the website, 3rc.org/MPS is where you can go for that. So yeah, that's all my slides for today, but I'm really happy to take any questions about us or our projects.
Really excited work. Thank you for walking us through that. We'll have a few minutes for questions. I know you said maybe it's in development still, but could you talk to maybe the number of compounds you're interested in, and then how that selection is maybe going?
Yeah, so we've asked the companies so far like what they would be willing to contribute and what would be realistic for an in-kind contribution. And I think we're landing pretty small numbers to start, like 4 compounds maybe that they're going to initially run. So we're just in early stages of trying to figure out, okay, well, if we only have 4, what exactly are we going to choose for? We're hoping to choose compounds that are not so obvious that like a very simple cell model would pick up toxicity, but some that are actually weren't necessarily just found and are, just have a little bit more gradient to actually showcase the ability of the MPS to pick up these signals. And we are going to do them at different doses, make sure that we have negative controls. So starting pretty small, but then after that, it'll just kind of depend on what funding we get and actually budgeting out costs. And so we're hoping to actually develop a like somewhat prioritized list. So like what are our top 4? And then maybe what are 20 additional that if we run a full study, what can we do there? And that's still in kind of early process. So Nicole and the end users are kind of taking the lead on that. And I believe they should be meeting soon to kind of discuss. Our companies have provided suggestions on various compounds, but we do really want it to be kind of independently decided. So yeah, that's where we're at on that so far.
No, that's great. And maybe you've heard some, but we've done a lot of curation efforts, both on the rat side and then, I know WIDA's involved, so you probably have the DILI list, but some of the other info there. So I think if there's ways we could support or have gathered some of the data, let us know.
Yeah, definitely. And Nicole said that, she has that list. And so I'm hoping that she can kind of be our main connection point. But if others want to be involved, or if you guys see other ways we could be connecting better with HESI, we're certainly happy to get more involved. So
great. Let's go to Carol.
Yeah, thank you very much. These are, obviously really exciting times. These systems is really going to change everything. I was curious about the compounds, but I'm also curious if could you say a little bit more about the endpoints that will be measured in the microphysiological systems? What predictive markers are you going to be looking at?
Yeah, so we're still deciding exactly what we're going to be doing. I'm actually going to pull up, so I'm actually quoting you right. But we are thinking a lot about like things like ALT, albumin, AST, urea. Those are some of the ones that the companies currently consider optimized for their systems and that they would recommend looking at. And so we need to actually make a formal decision about what outputs we're going to decide on. The other thing that was recently suggested by one of the companies that was an interesting suggestion, although we'd have to consider how we would design it exactly, would be, rather than maybe mandating a particular endpoint, actually looking at, more that are they seeing these reference compounds based on their optimized endpoint are falling into the right categories of like toxic or not toxic, etc. I'm not a data scientist expert enough to tell you how you would actually do that, but that was one suggestion. And so yeah, we're currently kind of throwing that around as an option because some of the companies really do feel like, well, our platform is optimized for this output. And so
that allows a little bit more of a head-to-head comparison with the clinical trial data. I was curious if you might consider throwing in some transcriptomics.
Yeah, and I think for the transcriptomics, I think what we've heard from the company so far is like that would be harder to do with the in-kind early study. But if that was something that, for more full-fleshed protocol, that they might be more willing to do that. So, yeah.
Because as you know, we've got lots of good transcriptomic biomarkers that we're developing in this committee that it would be really interesting to see how they perform in such systems.
Yeah, and that's like, I know originally when I connected with Connie, there was like the question of like, oh, like the cell painting, would it make sense to like layer this on top? And I think the big thing, at least for now, is and this is always a struggle, is like remembering like what's the core goal of this project, and it's not to really find new biomarkers or anything like that. It's about when the FDA sees MPS platforms fall on their desk, how do they know that it's like good enough, that it's validated, that that data is. So I think we have to keep trying to remind ourselves to keep it on the simpler side. But it is an exciting, like, I think there's so much potential to take what we do from this early project and expand in a lot of ways earlier, and just having this consortium of all these liver developers in one place, and just like us and the FDA, like all these partners, I think is really exciting. So we'll see.
Yeah, thank you.
Yeah. And Hydron had a good point in the chat that if there's a way to maybe save it, so maybe it's something, and we'd have to think about time points and what assays are terminal or maybe already lyse it, but that might be a way, and maybe coming back to us and seeing who would be willing and interested, and totally hearing you when we flesh out the protocols. And I think Hydron has some good comments on what type of buffer to use.
Okay. Yeah. Good to know. Yeah, I will say like a few notes on our things are like, we aren't planning on mandating a particular media that the platforms work on, because again, we're really trying to balance both letting the platforms be optimized for what they are, but then also actually having data that we can compare. And that's been quite the challenge, but one that we're kind of playing with. And again, thinking of that end goal, like the FDA is going to see a lot of variation in their protocols. So just like what's within the range of expectations. So
yeah, very exciting. I hope this is something, we can stay in touch and continue to get updates on, because I think it informs a lot of our work and next steps. So that's good.
Yeah, it's great to hear about OASIS as well. So we want to stay updated on what you're doing too. So thank you.
Great. Let's everybody thank Megan.